Welcome to The Explainer. Whenever you look around at the world, you just see this incredible diversity. From a towering tree to a tiny insect to you and me, life takes on an endless variety of forms. But what's the fundamental unity underlying all this breathtaking diversity? Well, today we're going to explore the absolute basic unit of life, the cell. Understanding this microscopic structure isn't just some boring biology trivia. It's the ultimate key to understanding disease, genetics, and honestly, the evolution of life itself. We are going to unpack the blueprints of how every single living organism is built and organized. Okay, let's dive into this. We'll start with number 1, the cell theory. Then 2, anatomy of a cell. 3, eukaryotes versus prokaryotes. 4, inside the cytoplasm. And finally, 5, cell shapes and sizes. All right, section 1, the cell theory, unlocking the mystery of life's fundamental unit. I like to think of the formulation of the cell theory as a classic 19th century detective story, right when scientists were just beginning to discover the basic rules of life. Our story actually kicks off in 1838 with Matthias Schleiden, who was a German botanist. He examined a massive number of plants and realized, hey, they're all composed of different kinds of cells that form plant tissues. Then, just a year later in 1839, a German zoologist named Theodor Schwann was studying animal cells. And Schwann made a huge breakthrough. He reported that cells had this really thin outer layer, which we now know today as the plasma membrane. Schwann also noted that having a cell wall was a completely unique character of plant cells. This led him to what was a truly radical hypothesis for the time, that both plants and animals, which look so completely different to us on a macro level, are actually built from the exact same microscopic building blocks. Together, Schleiden and Schwann basically pieced together the initial cell theory. But there was a major missing puzzle piece. How were these new cells actually forming? Well, it wasn't until 1855 that Rudolf Virchow stepped in to solve the mystery. Virchow provided the final, absolutely critical piece of the puzzle. He famously declared, omnis cellula e cellula. In Latin, this translates beautifully to all cells arise from pre-existing cells. Now, this wasn't just a neat little addition to the theory, it was a total game changer because it actively shattered the prevailing theory of spontaneous generation. That old misguided idea that life could just spring up spontaneously out of non-living matter. He explained that living cells literally divide to form new ones, finally giving the cell theory its complete, finalized shape. And this brings us to the cell theory exactly as we understand it today. It rests on 2 absolute foundational rules for all living things. First, all living organisms are composed of cells and products of cells. And second, all cells arise from pre-existing cells. Literally no exceptions. Just think about that for a second. Every single one of the trillions of cells currently operating in your body right now obeys these 2 exact rules. This theory creates such a profound sense of unity across every single living thing on Earth. Moving on to section 2, anatomy of a cell, crossing the boundary into the micro world. Now that we know the overarching rules, let's zoom in and look at the physical geography of a typical cell. If you've ever looked at an onion peel under a microscope in a biology class, you were looking at typical plant cells. If you looked at human cheek cells, you were seeing typical animal cells. Now, there are 2 key differences here regarding their outer boundaries. Plant cells have a distinct, rigid cell wall on the very outside, with the cell membrane situated just inside it. This cell wall is typically made of cellulose, and it provides massive structural support. It's literally what allows plants to stand upright without having a skeleton. Animal cells, on the other hand, they lack this rigid wall entirely. They rely solely on that outer cell membrane as their delimiting structure, which gives them the flexibility required for animal movement. When we push past that outer boundary and look inside, we find a dense, membrane-bound structure called the nucleus in each of these typical cells. You can think of the nucleus as the critical command center, the central vault of the cell. Why? Because it houses the chromosomes, which in turn hold the ultimate blueprints of life, your genetic material, DNA. The nucleus uses this DNA to strictly regulate gene expression and direct pretty much everything the cell does. Section 3, eukaryotes versus prokaryotes, the great cellular divide. That nucleus we just talked about, its presence or its absence actually forms the most fundamental classification system in all of cellular biology. Eukaryotic cells are those that contain a distinct membrane-bound nucleus. Prokaryotic cells, however, completely lack a membrane-bound nucleus, and this simple structural difference dictates the entire complexity of an organism. Having a nuclear envelope in eukaryotes protects the complex DNA from the chaotic, constantly churning chemical reactions happening in the rest of the cell. This evolutionary advantage is precisely what allows for vastly more complex, multicellular life forms, like us. Section 4. Inside the cytoplasm, the main arena of cellular activities. Let's step even deeper inside the cell now, moving past the nucleus to explore the surrounding space that fills up the volume. This space is called the cytoplasm. It's this semi-fluid matrix occupying the volume of both plant and animal cells, and prokaryotic cells too. So the crucial point is, this is where the action happens. The cytoplasm is the main arena of cellular activities. It's exactly here that vital metabolic pathways constantly occur. For example, the very first steps of breaking down glucose for energy, that happens right here in the cytoplasm, to maintain the living state of the cell. Floating right within that semi-fluid cytoplasm of eukaryotic cells, we find distinct, highly specialized structures called organelles. Honestly, you can think of a eukaryotic cell as a massive biological factory, heavily compartmentalized for maximum efficiency. The endoplasmic reticulum, or ER, serves as the primary manufacturing floor. The Golgi complex acts as the shipping and packaging department. Lysosomes function as the cellular waste recycling centers, and mitochondria, those are the vital power plants generating ATP energy for the entire operation. You'll also find microbodies and vacuoles doing specific storage and enzymatic tasks. But remember our great divide from earlier? Prokaryotic cells entirely lack all of these membrane-bound factory compartments. However, there are a couple of brilliant exceptions to the membrane rule that are really critical to understand. Ribosomes are non-membrane-bound organelles, and because they lack a membrane, they are found in all cells, both eukaryotic and prokaryotic. Ribosomes are essentially the protein factories, translating genetic code into physical action. In eukaryotes, they aren't just floating around in the cytoplasm, they're also inside chloroplasts, inside mitochondria, and attached to the rough ER. Another absolutely vital non-membrane-bound organelle is the centrosome. Found exclusively in animal cells, centrosomes are crucial because they form the spindle fibers that literally pull chromosomes apart during cell division. All right, Section 5, Cell Shapes and Sizes. The sheer diversity of life's building blocks. We've talked a lot about what's inside, but the physical dimensions and forms of cells are just as staggering. Think about this number, 0.3 micrometers. That is the length of mycoplasmas, which hold the record as the smallest known cells on Earth. To put that crazy scale into perspective, a micrometer is 1/1,000,000th of a meter. You could comfortably fit thousands of mycoplasmas side by side across the width of a single human hair. No way, right? Let's move to and see how this builds across the whole cellular universe. If mycoplasmas are 0.3 micrometers, typical bacteria sit around 3 to 5 micrometers. Moving up, human red blood cells are about 7.0 micrometers in diameter. Now, you might be wondering, why don't we have cells the size of elephants? Well, it mostly comes down to the surface area to volume ratio. Cells generally have to remain small to efficiently transport nutrients in and get waste out. But there are fascinating exceptions. The absolute largest isolated single cell, the egg of an ostrich. And nerve cells take the crown as some of the longest cells in existence. These massive cells are incredible, unique biological workarounds to that standard size limit rule. And this brilliantly illustrates that it's not just size that varies out there, it's shape too. Cells can be disc-like, polygonal, columnar, cuboid, thread-like, or even totally irregular. Consider mesophyll cells from a plant leaf. They are perfectly round and oval. Why? So they can easily pack full of light-absorbing chloroplasts while leaving small air spaces between them to absorb carbon dioxide super efficiently for photosynthesis. The big takeaway here is that a cell's shape is never just random. It is directly, physically tied to the specific function that the cell performs. Form always follows function. Which brings us to one final, kind of mind-bending thought to leave you with today. We know from Rudolf Virchow that the cell theory states all cells arise from pre-existing cells. That means the cells dividing in your body right now are the result of an unbroken, continuous chain of cell division, stretching back literally billions of years. So the big question is, if every cell comes from a cell before it, what did the very first cell look like? How did it form? It's a question that continues to drive the incredible science of biology today. Thanks so much for joining me on this deep dive explainer, and keep questioning the micro world around you.